Hello, and welcome back to Introduction to Psychology. Last class, we were talking about survey research. And let's say that we are gathering information about individuals. We can give them a self-report questionnaire, which is probably what's going to happen when you go and participate in research here. You'll get a survey and it'll ask you all kinds of questions about yourself, and you answer often on a Likert scale where your response is somewhere between 1 and 5. And it's you talking about yourself. It's about your self-perception. But there's ways that you can be biased. You might want to appear in such a way that it pleases the experimenter, that the experimenter likes you. If the surveys are given as part of, say, a job interview, well, then you might want to present yourself in a way that'll get you hired and not be honest about the things they might, not select you for if you were honest about them. So there can be bias in self-report. And so one thing we can do is also get reports from other people. So you can fill in a self-report personality survey, and we could also give surveys to your friends about your personality. And they'll be different, validly different, because you'll behave differently towards your friends. Doesn't mean they're wrong, but we get a better picture when we look at all these different sources. And now I see a question in the chat. And it's a question about taking part in research, participating in research in order to get course credit. You can participate in the research, or you can go and read papers and summarize them. So you can do one or the other or both. Now, back to ratings. So other people's ratings are not totally unbiased and objective either. And there's something called the halo effect and the horns effect. If we like somebody, we are likely to give them better ratings on socially desirable constructs than we would if we didn't like them. Put it this way. Imagine if there is let's say that we had a rating scale for, let's say, being a good student. I'm just making this up, but let's say it had a bunch of items on it. It's like a performance rating. And one of them is about the quality of your academic work, your grades. Another one is about how engaged you are in class. And another one is about how timely you are. And another one is about how well prepared you are. And another one is about whether you've done the reading. You get the idea, right? There are a bunch of different things. And say you could have really high academic performance, but come in late. Okay? Like these could be orthogonal or not related. And let me try to think of something else that could be really unrelated to other measures. Whether you get along with your classmates or not. Okay? Now, let's say that I really like you and you're the teacher's pet. Well, maybe you're late, but yeah, you came in the back. I didn't even notice. So we'll give you 5 out of 5 on timeliness, even though you don't quite deserve it. Somebody who is biased in your favor is likely to go down that whole list of totally different questions and kind of give you a 5 on everything. Right? Like when students who like me rate me on those student ratings, they'll just like give me 5s on everything, like even if one of those things is something that I'm not particularly good at. Now, let's do the opposite. Let's say that, let's take the example of a performance rating at work. Let's say your boss is rating you on your performance, and you've embarrassed him in front of other people. He's very sensitive, and actually he wants your job for his nephew, and you're in for it. Okay, he really doesn't like you. Well, now let's say that you're legitimately skilled at something. Well, he's not going to rate you as highly on that as he would rate his nephew on it, would he? And let's say that there's a lateness indicator. Well, that one time you came in 5 minutes late was actually incredibly disruptive. So we'll give you a 1 out of 5 on that. Okay, did you get the idea? When there's a halo or a horns effects, we see sort of a strong correlation between ratings on kind of completely different things that are maybe not really connected. But what we're seeing is kind of the positive lens someone's seeing you through, the rose-colored glasses, or they're seeing you through, you know, with horns on, and they don't like anything about you. I got there was an employee engagement survey at my other workplace where I'm not a very happy camper, and sometimes I was reading the questions, I didn't really understand them. I'm like, I'm not sure what you mean by this. And maybe the fair thing would have been to give them a 3, right? Like 3 out of 5. Like, you know, if there was no I don't know or I'm not sure, it depends, but I guess 3 out of 5 is the middle. But I was kind of ticked off at them and kind of angry, and I just gave them 1s. Sometimes without even fully reading or understanding the question, because I just wanted to communicate my sense of dissatisfaction. I see some questions in the chat about experience. Looks like it's sorted. Okay. Correlational studies are a way of quantifying, studying, capturing, measuring, quantifying the strength of a linear association between 2 variables. And so there's some scatter plots here. These are called scatter plots. And you can see that on the x-axis, we have number of beers. And I think it's very small. I can't quite make out the font, but I think the y-axis is like your grade. And the idea here is, or yeah, like maybe grade on a midterm. And what this relationship is showing, if you step back and kind of eyeball it, is that the more beers people drank, and each point here is a person. So this guy got a 75 and he drank 2 beers. And this person got a 70 and had 4 beers. Okay, so the general trend there is that the more beers people had, the lower their grade. That line there is called a regression line, and there is a mathematical equation that you would compute in order to get the slope of that line. The closer the dots fall to the line, the stronger the relationship is. If every single dot fell on that line, you'd have a perfect correlation of -1. But the strength of the correlation is in the value of that number. So a negative correlation of 1 is a stronger association than positive 0.5, right? You look at the absolute value of that number. Okay, so that's a negative correlation. And then some of the points fall closer to the expected value, like this guy over here, then, oh no, you can't see my laser. I just realized that I need to click this in order for me to get where's my laser pointer? Here's my laser pointer. Okay, so the closer these fall to the expected value is what's predicted, what would be right on that line. It's a prediction line. Kind of the smaller the error in our model, in our correlational model. But then some people fall like pretty far from the line. So let's say that we were talking about outliers. Okay, so this guy has drunk a lot of beer, but has gotten an unexpectedly high grade. Okay, this distance is like error in our model. We predicted that he'd be there on that line, and nope. Okay, so that space there is error in our correlational model. And outliers like that, or points that generally fall further from the prediction line, indicate a weaker relationship. Now, here is an example of a positive correlation. And there's more scatter, I think, in this relationship than in that one. And in the positive correlation, as the value of X goes up, the value of Y also goes up. So the higher percentage of classes attended, in general, the better the midterm score of the students. So it's a positive correlational relationship. I can see an outlier over here, right? This point falls pretty far from the line. And in that case, the person attended most of the classes and got a lower grade than we would expect for somebody that attends that much class. Here's an example of like no correlation, a correlation of 0. And in that case, you don't see a linear relationship. I don't think they should have even put a line through that. The association there is between shoe size and grade, and there's no relationship. So no relationship, this kind of looks like a cloud. However, the correlation coefficient is a measure of a linear relationship. And if you calculate a correlation coefficient for a U-shaped relationship, it will come up as 0. And so not all relationships between variables are linear relationships. Some of them are nonlinear relationships. Example of a nonlinear relationship is the upside down U-shaped relationship between performance and arousal. If, let's say, we're looking at your performance on the midterm, if you are like super relaxed and even drowsy and taking your sweet time, maybe you won't get such a high grade because you're just, you're too relaxed. But let's increase your arousal and your stress a bit, right, to a moderate level. And then, you're ready and you're alert and you're engaged and you're taking it seriously and you're pushing it through and you're going to get done, right? Maybe that's where your highest performance is. But then let's keep cranking up your stress and your arousal and your anxiety. And now, right, you're sweating and you're trembling and your heart's in your throat and you're panicking when you read the question and you're forgetting everything and you can't pull anything out of your memory anymore. Okay, now you're too aroused and you'd get a lower grade. And if that happens to you, please do some deep breathing. Count to 10. That'll help get your arousal down. We have brains that are geared towards seeing correlations. And I wish they would say association instead of correlation, because when you say the word correlation, you're talking about Pearson's correlation that's linear, and in fact, there are non-linear associations. So what they really mean here is association. We do have a tendency to notice that if two things co-occur, like once, we run with the conclusion that these variables are connected. So apparently, people who have joint pain when it's rainy might believe that that there's an association there, and the next time it's rainy, that you might even notice your joints a little worn. Say your biases could work there, but there isn't really any connection. This is a source of superstitious behavior. So athletes, especially in baseball, have a lot of superstitious beliefs and behavior. So let's say that before I had a really big win, or I did something really, really amazing on the field, I ate chicken, or my wife was sitting in the bleachers wearing pink. Then I might believe that that was the reason I got lucky and try to keep repeating that. Try to always make my wife sit there in a pink shirt. Try to always eat chicken. Maybe one day when I had something bad happen, I noticed something a little unusual, like a black cat crossed my path. Well, now I'm going to be afraid of black cats crossing my path. See how the human mind just has a tendency to do that. See associations. And there's a reason for that, and it's because we notice when things are connected because we care very much about our outcomes and we're interested in what predict good outcomes or bad outcomes. So we have a bias towards seeing these connections. But what we don't notice is non-events, right? All the time something didn't happen. all the times you had a bad day and there were no black cats involved. You might have experienced this phenomenon. Let's say that you think about your friend, and then your friend texts you and you're like, wow, it's telepathy. That happens to me. I'll think about my old boss and then she'll send me a text. I'm like, oh, we have this connection. Really, I'm noticing those two events and running with that idea. But there have been many other times that I've thought about Lynette. She gave me my briefcase, so every time I see my briefcase, I think about Lynette. Right? There's many times when I think about Lynette and she hasn't texted me. But we don't notice those non-events. And if we did, we'd realize that there was no special connection between the two positive events. As we talked about last class, just because there's a correlation between two variables doesn't mean that there's a causal association. There might be, right? In some cases, it's more rational to speculate that there is a causal relationship. If there is a causal relationship, the correlation itself doesn't tell us the direction of causation. So let's say that you have variables x and y have a correlation of 0.5. Well, the correlation between x and y is 0.5, and the correlation between y and x is 0.5. And there's nothing about that tells you whether x causes y or y causes x. And as we were talking about with that ice cream example, the correlation between ice cream purchases and shark attacks or violent crimes, it's not necessarily the case that ice cream consumption causes people to act more violently. The heat is a third variable that increases both ice cream consumption and violent crimes and shark attacks. But what correlational studies do, just as case studies and introspection, is that they give us a hypothesis, right? They give us an idea, and then we can go and test that out. We can design more controlled experiments to test the idea. So the only kind of research design that can establish causation is the experimental design. And there are controls that we need in order to do a good experiment, because just because we did an experiment doesn't mean we did it well. There's lots of ways for bias to creep into experiments. So what's key about an experiment is that we are manipulating one variable. In a correlational study or a case study, the researcher doesn't intervene, right? You're just observing. But in an experiment, the researcher actually manipulates something. And that thing that we manipulate, right, the treatment that we do or do not give you or give you in different doses, is called the independent variable. So let's say I wanted to do an experimental study on a kind of therapy. I'll call it critical liberation therapy. It's where I convince you that actually there's nothing wrong with you. That'd be different for psychology. So I would have two groups because you need a comparison. And one of the groups of people would get my therapy and the other wouldn't. And if the, well-being scores were higher in the treatment group compared to the control group, it would suggest a causal effect of my treatment. There's still a lot of confounds, right? And psychologists do animal research where they can, because you can keep the animals in their cages in the lab. But maybe some of the people who were in my treatment group actually also had other things happen to them. They they got jobs. I don't know, right? There was something else that improved their well-being. So how do we control for that? Well, we try to get large samples because that spreads out the effect of chance things happening. When you have, let's say I only had 2 people in my study, 1 person in the treatment group and 1 person in the control group. Well, in that case, so many things could happen to that 1 person in the treatment group and the 1 person in my control group that maybe the difference had nothing to do with my therapy. But if I had 100 people, well now, chances are that overall, some of them will have some good things happen to them, and some of them will have some shit happen to them, and it'll be kind of a wash. When we get our sample, ideally it's a representative sample, but you're never going to do a study where you get to randomly select the participants from all of humanities. The studies that are being done here at Mount A are convenient samples because we tempt you with a research with credit towards your class, and that's why you're going to participate in it, right? And so the people who are doing the study, who are the subjects of the study, they're not a random sample of people in Sackville, New Brunswick, let alone people globally across the world. It's a very specific population, okay? It's called a convenience sample, and it limits the generalizability of the results. It limits the external validity, because maybe what we find, right, even if it's a well-conducted study, just describes folks like you. But we hope that it's more generalizable than that. When, let's say I have 100 people in my study, and I'm going to put 50 of them in the control group and 50 of them in the treatment group, the best way to do that is through something called random assignment. And it means that you're just as likely to be assigned to the control group than to the treatment group. Everyone in that sample is just as likely to be assigned one way or the other. It's like we flip a coin, fair coin, to decide which group you're going to be in. Nowadays, we don't do coin flips or picking names out of hats anymore. We use computer programs to randomize people. But that gives us our best chance ever of creating groups that are equivalent to each other at baseline. A really bad idea would be to say, take all the most struggling people and put them all in the treatment group, because that way they'll get help. Because then when we looked at differences, let's say that well-being was way higher in the control group, and I put all the sickest people in the treatment group, and let's say that the treatment worked, it would work, it wouldn't work as well on them because they're the most they have the most severely disordered behavior, and so I get a smaller difference than I'd expect. So you don't want to do something like put all the female people in one group and all the male people in the other group, or just use random assignment, because even though that doesn't guarantee that the groups will be equivalent to baseline, like you could through dumb luck get all the females in one group and males in another group. That could actually happen. It's very unlikely to happen, and it's also the best way that we have to control for variables that could be confounds that we don't know about, that we haven't thought about. Usually we'll think about some variables that could confound the study, like, oh, okay, could be age, could be gender, could be this, and you might have 5 of them in mind. You want to check that these are equivalent at baseline, but there's like 1,000 other things that could influence your study that you haven't thought of. And so by randomly assigning people to group, you cover those bases as well. I see there's something there, the operational definition and the dependent variable. The independent variable is the variable that you're manipulating. So in my little example here, it was treatment, whether treatment or no treatment. And then the dependent variable is what we're measuring. It's the outcome we're interested in that we think is being affected by the independent variable. So I think that if I give you treatment, that'll pull your score up. Whereas if you didn't get the treatment, we left it where it was, or if I give you horrible treatment, it'd get worse. There's this idea of kind of like pulling a string. That's why I think of it as the dependent variable. It's the one that gets pulled. So your well-being score is the dependent variable because it's getting pulled up by the independent variable, or pulled or pushed down. Okay. There's a question. Would an experiment ever separate men and women, then randomly assign them to two groups so that you have equal amounts of men and women? Would this still count as randomized? So the only thing you controlled was that there would be even amounts of men and women in both groups. Yes, that's a type of design that's called, I think, blocks, blocking. That is a method that can be used. Sometimes that method is appropriate. Let's say that you know that gender is a major factor that influences things in certain ways. So we'll say, okay, let's look at this study, this treatment in men and in women, and now we'll, within those two groups, randomize them to treatment or no treatment. So yes, that is that's a legitimate method. We need to define really not just the dependent variable, really everything you're measuring should have an operational definition. So if I am, I need an operational definition for what my therapy is. Okay, what does it consist of? How many sessions are there? How long are the sessions? What are we talking about? So I need to be clear about what that therapy is. I need to be clear about what the control is, right? When I'm saying control group, you're thinking, okay, what happened to them? Because let's say that it's a no treatment control. That's a type of control. That means that I'm looking at the effect of this therapy versus just leaving people alone. And that can be problematic because how do I know that my special therapy is any different than just giving someone attention? Because if people are sad and you take an hour to talk to them about their lives and seem to care about them, that can really perk them up. And, you know, that doesn't involve any kind of psychological psychotherapy, right? Any specific technique. So, you know, a better control might be a placebo group, okay, where what I give them is the psychological equivalent of a sugar pill. So you meet with somebody and just chat, but you don't do anything that would really look like it was theoretically look like it was this form of therapy. It wouldn't get at the kind of issues that the therapy is trying to get. And I would expect to find a smaller difference in the treatment and the control there, because it's getting rid of that attentional, that effect of giving somebody attention, that placebo effect. And then I would also need to have a clear operational definition of what I mean by well-being. And that's a really big philosophical idea. Well-being just basically means good being, living in a way that's good. What is good? According to what value system, what moral philosophy? So some psychologists like have measures that get at a hedonic idea of well-being, which is the idea that well-being is feeling pleasure and avoiding distressing emotions. But other psychologists might have what's called the eudaimonic idea of well-being. That well-being means that you are kind of self-actualizing, you're growing. So you can be in pain, that's okay, you just have to be growing. And then some other psychologists might go for a utilitarian vision of well-being, and they'll say, you know, what matters is that you can cope with the stress in your life, You know, it's not that you don't have stress. It's not that you don't feel distress. You just need to be able to keep going. You need to be able to keep contributing your talents to your community. Those are like 3 totally different definitions of well-being. So I need to be clear about what I'm measuring so I can communicate that. And so when you read the paper, you know what I was doing. That way you can replicate it. You can understand it better. Experimental designs can have, well, there's 2 main designs, between group and within group. You can actually combine them, but let's not get into that. In a between group designs, there are 2 non-overlapping groups that are given different treatments. This is like the treatment group versus the control group. And then we go and compare the difference between the groups. The difference between the treatment group and the control group. In a within study, and in a within group comparison, we actually do a pre-post kind of test. So everybody gets the therapy and we control their, and we measure their well-being before and after. And I'd be hoping that your well-being score is higher after the treatment than before. And that controls for some things, but not others. When in a between group study, you're comparing different groups of people. There's different people in the treatment and the control group. And there's all sorts of other ways that they're different from each other that could interact with the treatment effect. There's more variance, there's more error in doing that. When you do a within subject study, each person sort of acts as their own control. It's your score after the treatment minus your score before the treatment. And comparing you to you involves a lot less variance. So there's an advantage to a within group study, but there's always disadvantages. And how do I know that your score, your well-being increased because of the therapy? Maybe everyone who signed up for my therapy was really sad. They were kind of at the bottom of, you know, they were doing badly. And I gave them this therapy and they improved, but I don't know whether that's a placebo effect. And I don't know whether they would have just gotten better on their own without me doing anything at all, as a kind of regression to the mean thing. But you can always, you make more and more elaborate study designs to try to address these problems. So one way that experimenters have tried to control for this, I'll give you the example of school bullying programs. We'll give you a bullying program in grade 4, look at student scores before and after the program. We could maybe compare that difference to students who were in grade 4 last year and didn't get the program. So I'm not asking, this is not something you have to memorize for the test. The point I'm trying to communicate is that there's always gaps in a research design, and you can keep designing different studies and more elaborate research designs to try to get at the problem in the last step. A confounding variable is something that confounds your study. It messes up your interpretation. So if I was doing this study on my therapy and I put all the saddest people in the treatment group, that would confound my experiment. Because now the 2 groups, right now there's a difference between the experimental and control group, and it's based on severity of mental illness that affects the outcome. Ideally, the only way that these 2 groups are different is that one of them's getting the treatment and one of them's getting the control, and ideally they're same equivalent at baseline on all of their characteristics, but that never happens in practice. But if I did something like put all the most ill people in the treatment group, that would mess up my interpretation of the results. Because what would probably happen is that the treatment wouldn't work as well on people who are more severely ill, right? And that would sort of depress the effect of the treatment. We talked about placebo effects, where you get better because you're getting some attention. And that happens a lot in psychology. Your textbook says that 80% of the effect of antidepressants is attributable to placebo effects. So a placebo is when you seem to get better because you believe that you should be feeling better. You know, you're taking care of yourself, you're taking this little blue pill, you're going to your appointments, and you know, things are starting to look sunnier for you. And there's all these psychologists that are interested in your well-being. You get the idea, okay? The opposite of that is called a nocebo, and that's kind of like a voodoo doll effect. If you believe that someone could be harming you, you could really start to feel bad because of that. And an example I'll give, and it's one, you know, since we're not a culture that does voodoo, the closest example I can give is sort of school bullying or workplace bullying. Bullying is fairly common in workplaces, and the last time it probably happened to you was in middle school. But then, you know, when you go out into the workplace, it kind of can go back to being like that. If people have spread rumors about you, if there's gossip and rumor spreading, you never know when they're doing that, and you're always watching your back, and it's super stressful. You know, and then that stress can actually legitimately make you sick. So even though placebo effects are in your head, they actually can legitimately help you, and they can legitimately harm you. So, you know, people who are going through workplace bullying act like they're paranoid. They're always watching their back. They're keeping records of everything. They're being their own lawyer. Their stress is through the roof, and they start to get run down. Maybe they drink some wine in the evening, and they can't sleep as well, and they can just like really start to get hurt, you know, psychologically and also physically. With placebo, there's going to be a neurochemical level at which those effects take place. So with something like that voodoo doll effect or the bullying effect, that's increasing your stress. You're going to have a lot more cortisol, other stress hormones, and that's going to wear down your system. And then you're going to start to fray and snap. You get the idea. Like there's some biochemical process behind that psychological process. Same thing with placebo effects. So when you feel hopeful and optimistic, that affects your dopaminergic system. And so placebos have their best effects on conditions that are somehow associated or that rely on the activity of that dopaminergic system. And one of them is Parkinson's. Interesting question from Mika that popped up in the chat. Would internalizing messages given to you by bullies also be part of the nocebo effect? So if they're saying that you're worthless and you start to think that, interesting question. Just I need a minute to think about that. That sounds like a direct effect to me, right? That's a real harm they're doing to you directly. They tell you you're worthless, you feel worthless, you feel bad. But there's a part about being socially bullied where it's actually in your head. They're not doing anything, but you're going crazy because you never know that they might be doing something. They might be up to something. You're seeing plots everywhere. placebos work best on these conditions that respond to, let's say, the neurotransmitter system that's involved in a placebo effect. So things that respond to this idea of hope, this just dopaminergic activity in the reward center. So depression and chronic pain and anxiety are very, very subject to placebo effects, which is a good thing, right? If you have depression, if you have chronic pain, then you can cheer yourself up. You can change your outlook. There are a lot of things that can make you feel better. Now take the example of a type 1 diabetic that doesn't have any insulin. We know that placebos work better at higher doses. They work better when they're injected rather than given as a pill, because that way the placebo gets your bloodstream. You believe in it more. If you had a type 1 diabetic going into ketoacidosis and you gave them an injection of saline, you're like, there you go, it's your insulin. It's not going to work, right? That's, even if that person feels hopeful and relieved, it's not going to change their level of ketones. Other pitfalls in experiments. Researchers can bias an experiment by being too involved in it in a way that their hopes and expectancies actually change the result. So let's say that I really believe in my new therapy and I hope that it's a breakthrough and that it works and helps people. And I'm the one that is doing people's well-being assessments. And I know that Susie is in my treatment group, and I know that John is in the control group. Well, I might be motivated to perceive Susie as having better well-being. In fact, I might treat her differently. I might say, hey, Susie, great to see you. Beautiful day, isn't it? And maybe she'll react to me in a way that that'll perk her up, and that will legitimately give me higher well-being scores, or cause me to give her high well-being scores. And now let's say I know that John is in the control group. He's not getting my amazing therapy. Well, then maybe I won't treat him that way. Maybe I'll be a little colder to him. Right? Maybe maybe we won't have so much conversation. Maybe I won't give him that big grin. And maybe at the end of the day, I'll rate him as having lower well-being than Susie, right? I mean, they might even act the same way, but if I know that Susie's in the control group, it might be that in the treatment group, it might be like that halo effect thing. I'll think, oh yeah, she's wearing brightly colored clothing, and right, I would see it because I want to see it. Demand characteristics are cues that the experimenter gives the person on how to behave. The experimenter is in a position, or the researcher, the psychologist, the scientist is in a position of power relative to the research subject. And, people who are in subordinated positions often want to please, or they kind of follow cues. And so the researcher can give, even unconsciously, cues to how the person's supposed to behave, and they'll follow along. And there's, some major stories or phenomena that were really just caused by demand characteristics. One of them is hysteria. Psychologists used to believe in hysteria, but what the psychologists were really doing in that case was kind of coaching the women on how to act hysterical and faint, and rewarding them for acting in that way. And then showing off these patients at medical conferences. There's an example in your textbook of Clever Hans, the horse. And there was this horse that seemed to be able to do math, really complicated math. And he'd give the answer by tapping his hoof. So he would strike his hoof 4 times if the answer was 4. But what was actually happening was that people were kind of screwing up their faces when they got to the right answer. So if the answer was 4, that people were and the horse was picking up on that. So this table summarizes the different kinds of research studies. And I don't want you to think that, you know, some type of research are better than others. We need all of these different kinds of research studies, and they're appropriate in different settings and situations. If you want to study feral children, like what happens when a child is raised by a pack of wolves instead of human parents, you don't want to do an experimental study on that because that would be unethical. But you couldn't even really do naturalistic observation because you'd be required to intervene. If you find a child in the woods being raised by wolves, you can't sit there with a clipboard and be like, let's see what happens. You'd be required to go and take the child away. But you could do a case studies, right? You can find kids who you can find feral children who were saved and then, usually institutionalized, and you could go study them and try to talk to them and their caregivers. That would be appropriate. So you can't say, oh no, that's the wrong kind of research. Go do an experimental design to see if there's a relationship with causation. Get the idea? So when we're studying something, we want to use the most appropriate research method. And we're also constrained not only by ethics, but also by costs. Experimental studies are really expensive. And that's one of the reasons we do a lot of correlational studies. We don't intervene when there's a correlation, but it's easy to give 200 people a questionnaire and get them to fill it out. And so they all have advantages and they all have disadvantages. And ideally, when you're researching some phenomenon, you'd be able to find some studies that are case studies, that are naturalistic observations, that are correlation. And then there'll be a few experiments that can speak to whether there's a causational relationship. So they all have their place. I feel like this could be a good point to stop at. So next lecture, we'll cover the remaining slides on ethical guidelines for human research. There are also ethical guidelines for animal research. So I'll stop the recording here and I'll be available for questions.